Right, April?
Yes.
That's good. And let's start. Greetings, everybody. Welcome to the Bioinformatics seminar. Today, I have the honor to introduce Dr. James McInerney. Professor McInerney is an Irish-born scientist and former head of the School of Life Sciences at the University of Nottingham. He holds the chair in evolutionary biology and is a principal investigator in the School of Life Sciences. In 2020, he was elected president of the Society of Molecular Biology and Evolution, the world's largest evolutionary biology society. Professor McInerney was elected as a fellow of not only the American Academy of Microbiology, but also the Linnean Society. Professor McInerney's research group has contributed significantly to the current understanding of horizontal gene transfer, eukaryotic origins, and the topic that we are going to learn about today, the origin and maintenance of prokaryotic pangenomes. Now, if you are available, please open the microphone and unmute yourself and join me to welcome Professor McInerney. Please take the floor away.
Okay, thank you very, very much. It's a real pleasure to be here. Well, I'm obviously here in my front room of my house, but be here virtually as well in this meeting. I'm going to take you hopefully on a little sort of a journey. So some of what I'm going to talk about at the very, very beginning is oh dear, I can't find how to put up my slide. Oh, here, I got it, found it. Okay. Let me know if you can see things. Can you see my slides?
Yes, we can.
You can? And if I put it up now, can you see them?
Yeah. Yes.
Perfect.
Lovely, lovely. Okay, right. Brilliant. First part of the talk, success. Right. So what I'm going to talk to you about a little bit is I'm hoping to take you on a small journey, both in terms of how we've thought about genomes and maybe where we're going to think about them into the future and what our our understanding recently of them, what this means. I'm going to talk to you about prokaryotic genomes today. I know probably a little bit more about them, but some of this might actually be useful as well when thinking about eukaryotic genomes. I'm going to start off with sort of, first of all, recognizing the people that in this particular work, or in this particular set of slides that did all of the work: Maria Rosa, Fiona, Rebecca, Nadine, Martin, and Alan. Alan is with me right now. I haven't made the font of his name any bigger for any reason other than I think it's Microsoft are trying to make me do things that I don't want to do. So they did all of the work, they did all the programming. My lab is entirely bioinformatic, and it's entirely devoted to analyzing genomes and trying to understand what these genomes mean. So I'm putting this slide up as a sort of a slightly provocative first slide because it sort of maybe ties together all of these ideas that I'm going to try to talk about. The hypothesis is that evolutionary history is tree-like. And so for many people, this isn't just a hypothesis. It's, it's how we think about things. We think about evolutionary history as this great trunk of life that preceded the diversification that we see today, and then this trunk gives rise to branches, and branches give rise to smaller branches, and all the way to leaves, which are the current species today. And people sort of maybe highlight things like Charles Darwin's notebook, where he drew a figure of a tree and said, I think, and it's a branching diagram that shows that. But I mean, very, very interestingly as well, if you do read Darwin, he also allows for evolution to be non-tree-like, and he talks about certain species that might be hybrids, and he talks about hybrids quite a bit. So hybrid evolution is not quite that tree-like sort of endlessly diversifying evolution that we might think about. So for me, this still remains a hypothesis, and I'm possibly going to try to persuade you today that this hypothesis has been falsified. And not just that, but thinking about things as a tree, think about evolutionary history solely as a tree, might prevent you from seeing some other things that are very, very interesting. But I don't prevent you from thinking about tree likeness in parts of evolutionary history. Certain things have diversified and have had independent evolutionary lineages and evolutionary history, but sometimes evolutionary objects fuse together and merge, and that's really important for evolution and for life. I'm going to maybe just first of all talk to you about some of the things that we've talked about in this and ways in which we've thought about it. Here's a paper from about 10 years ago. We published this where we're just really talking about how certain genes really, and this is at the gene level, certain genes really look like these sort of matryoshka dolls. You know, you've got bits of evolving objects within bits of evolving objects within bits of evolving objects. So you can think of protein domains in a multifunctional protein. That's maybe quite often you see fusions of multiple multifunction proteins. And we started drawing these genes and these proteins as nodes on a network, drawing connections between them. If we can make a statement of homology between the genes, so that's what these lines mean, these edges, and we've colored them according to how, if you like, hybrid we think they are. So the ones that are green, we can't really find any relatives outside of just sort of this group. They don't seem to have undergone much recombination, much sort of non-tree-like evolution, when all the way over here to these red ones here, where we are finding genes that we think have very, very diverse evolutionary history and have been put together sort of like Lego pieces. We think this is a very interesting thing. And about 10 years ago, this really got us thinking about what happens if you start to throw away phylogenetic trees just for a minute, not forever. They're really good things. But how about we start trying to look at evolution without really looking at it in that way, just through that lens of phylogenetic trees. These networks are one way of doing it. We have lots of other ways of doing it as well. We started seeing horizontal gene transfer, or what to borrow a phrase from a friend of mine, Eric Baptiste, where we see the merging of evolving objects with one another. So protein domains evolving together, to make a new gene, two genes coming together, genes transferring from one organism to another to come together. All of these kinds of merger events. And once you start looking for them, it turns out you find quite a lot of them. TB. People said that TB doesn't undergo horizontal gene transfer, but when we got some TB genomes and we started looking at the evolutionary history of the mycobacteria, the Mycobacterium tuberculosis complex, we see that in the history of TB, we see horizontal gene transfer. So this is a very central part of Mycobacterium tuberculosis. It's metabolism. It's where this circle adds on these two carbons onto a growing fatty acid chain, and Mycobacterium tuberculosis makes all kinds of splendid fatty acids, and all of those pink symbols there. These are functions that show a history of horizontal gene transfer. So even in organisms where people have historically said there's no horizontal gene transfer, not much, we're still finding some when we go and look at it. Moving along, and this isn't really Pangenomes, but I'm just trying to establish the fact that as we looked more and more and more at different places and different kinds of organisms and so on, we saw more and more and more, we saw horizontal gene transfer and having an influence. These are 4 papers. This one here, acquisition of 1,000 new bacterial genes physiologically transformed a methanogen at the origin of haloarchaea. So there are these methanogens, they're archaea, and it looks like the halophiles, which are aerobic, heterotrophic organisms that do things with light, and their ancestors were anaerobic chemolithotrophs, the methanogens. And so this massive transformation, physiological transformation, all facilitated by horizontal gene transfer. And indeed, if you look at lots of archaeal clades, they have had huge influxes of genes from bacteria. Here, these bottom two papers talk about the origin of the eukaryotic cells. So prokaryotic cells, relatively simple, don't have a true nucleus. Sometimes they have inclusions in various, membrane-bound little organelle type things, but they never reach the kind of complexity that eukaryotes reach. And we think that's a simple merger of two kinds of prokaryotes, an archaea and a bacterium, to form eukaryotes. And we've, again, we're looking at the merging of these genes together, making new organisms, establishing that evolutionary history is sometimes, and really quite often, not very tree-like. You know how you see in the textbooks phylogenetic trees representing the evolutionary history of life on the planet? Well, this is another way of looking at it. It's orthogonal to that sort of phylogenetic tree that you see. But this is going back to our genes and our gene families. And we've made communities here, and we've colored the communities according to how many hybrid genes are in those communities. And the connections, again, are statements of homology, regions that are homologous between two gene families, and we just draw an edge between them. And there are something like 3 million genes in this, so we've had to represent some of them as sort of larger communities. But the more green-like it is, the less you see in that community hybrid genes, hybrid proteins. And the more red-like it is, the more hybridizations, and the more you're seeing these, genes within genes within genes, or parts within parts within parts. So I'm just going through all of that. I'm not going to go into any details there. The methods, and I realize I'm talking to a bioinformatics group, mostly BLAST, all versus all BLAST searches, making networks quite simply by joining two genes or whatever we're looking at, two parts of genes together. If they show homology to one another, we do this. And then using network mathematics, we make communities. We start trying to cluster these things together in order to get to visualize them and to understand what's going on. But the take-home message from, I guess, the first of these slides is that once we started looking at a non-tree-like evolution, we did find it quite a bit, and we found it in a lot of places. And so maybe what I wanted to do today is to maybe say, how this happened, what had happened, and so on. So this is kind of a picture of, if you like, a fairly big pangenome. It's the pangenome of archaea. And over here on this side here, so I didn't throw phylogenetic trees away completely, is a phylogenetic tree, a backbone phylogeny of a whole bunch of archaea. And all of the different groups are colored here, or they're highlighted in alternating light gray and dark gray here, just so that you can visualize them. On top here, this thing that looks like a sort of a Manhattan plot or a Manhattan skyline, these are individual bacterial genomes, and the height of the line just represents how big the genome actually is. Okay, and they're ordered in a particular way that makes a little bit of sense for us to visualize. The cells in this matrix here are more red if they're more bacteria-like. So the cells are, for any gene that's in, say here, methanosarcinales, how archaea-like is it? How much is it like the rest of its cousins, and how bacteria-like is it? And so it's red here because this gene shows more similarity to bacteria than it does to other archaea. So what we are seeing are large floods of genes into archaea from bacteria, sometimes, not always, and some bacteria seem to be more involved in these transfers, and some seem to be less involved in these transfers. But as I said earlier, I put up a slide of this, it physiologically transformed, in particular the haloarchaea here. But when we looked across the whole archaea, it's had quite an effect on these, and quite a lot in particular of their metabolic genes are genes that they've stolen from somewhere else. It's a very non-tree-like sort of evolutionary history. We expanded this out to look at lots and lots of archaeal groups. I won't get into this, but each of these chunks of genes here, these are archaea here again, bacteria along the x-axis, different ones, different import families we call them. And these chunks here are unique to either one group of archaea and a bunch of bacteria, a group of archaea and a bunch of bacteria, a group of archaea. So in other words, these genes are in a particular group of archaea, but not in their close relatives, or what we think are their close relatives. These are all imported from elsewhere. So again and again and again, we're seeing this sort of thing happen. The origin of eukaryotes was a major merger again of things. And here, just a little diagram from a kind of perspective article we wrote. Archaea and bacteria contributed to the origin of the eukaryote, leading to eukaryote cells today and mitochondria and so on. So just wanted to talk about that first of all, to establish what we think actually is happening in life, in certainly microbial life, quite a lot. And we worked on that maybe from the year 2000 to 2010, 12, 14, and we thought that was all really interesting. But something happened when people sequenced the third E. coli genome. So the first one comes out, it's a major milestone. Surprisingly, E. coli wasn't the first genome to be sequenced. It was about the, I don't know, third or fourth or fifth. And then the next E. coli, and it wasn't terribly similar to the first one, but okay. And then the third one comes along. And there's this remarkable paper, it's now 16, 17 years old, where they said, look, when we look at the genes that are common to all three E. colis, It's less than 40%, 39.7%, something like this. The rest of the genes are either in one or in two, but not in all three of the E. coli's. Think about that. If you met three humans that only shared 40% of their genes, one of them would look very much like a starfish. Okay? They would be that different. But every microbiologist, every card-carrying microbiologist would look at these three E. coli and say, yeah, they're E. coli. They really are E. coli. We had a new problem. And the problem was there's too much variation, right? There's just too much. And so we sequence multiple genomes of multiple species, and this view emerges of what we now call the pangenome. And this is a little sort of Venn diagram type thing that we put into a paper a few years back. And just to try to illustrate it, in the center, you've got the core genome. These are genes that are common to all isolates, all sequenced genomes that we have, and the accessory genome that it's common to fewer than all. And the pangenome is the union of the core genome and the accessory genome. So then that's fine. So we spin along for maybe a decade, and lots of people sequence lots of genomes and report what's present in one strain and not in the other, and present in the first and not the second. But it struck us about 5 years ago, we need a mechanism. We need to explain this, not just in terms of, the nuts and bolts, the description of what the pangenome was, or the description of for Salmonella, it looks like this, for E. coli, it looks like this, for Yersinia, it looks like this. We need to say why it exists, because there's just too much variation. I'm just very briefly going to digress a little bit. Science found too much variation around about 1966. People were working on isozymes of proteins. And until then, we just thought that natural selection probably meant that genes and proteins were close to perfect, might be a mutation or two, but these genes were, would probably show very little variation. In 1966, Dick Lewontin and Hobby published a bunch of papers where they said, look, there's a lot of variation. We can run these gels, these starch gels, and we look at variation in different kinds of enzymes. And they're not all the same. Within a species, you've seen quite a bit of variation. Maybe even within an individual, they'll have two alleles of the same sort of gene, and they're producing two different proteins that are that little bit different. That led to the neutral theory of molecular evolution. Within about a decade, people were really quite convinced that a lot of variation in genomes was neutral, that you could substitute one amino acid for another. It probably didn't matter all that much, or that when we saw these substitutions, it didn't matter all that much. So I digress there just to say that the neutral theory is well established. Though everybody would also say that natural selection is a very, very strong player. So it struck us that we should sort of make some kind of statement about why we thought that pangenomes existed. Do they exist because, on the one hand, it's just variation? It's just variation. Organisms can tolerate variation. The mutation rate causes variation. If you've got large populations, that probably creates a lot of variation. And natural selection, if two things are almost the same or pretty much the same, natural selection doesn't actually prefer one over the other. So I'm going to get to it in a second. We were faced with three ideas for why pangenomes exist, why we're seeing the data that we're seeing. I just want to maybe digress as well to say it's slightly unusual in science to, first of all, get data and afterwards have to sort of somehow retrofit theory to it. This was unexpected data. Quite often in science, if you think about the Higgs boson, for instance, right? Many decades ago, Higgs said there should be a boson, there should be this sort of element. And it took decades before he was proved to be right. So the theory came first, and the data to test that theory and to prove it to be correct came later. But here we had data, and we didn't really have theory that fitted the data. And so we had to think about how we were going to think about these things. So what about purifying selection? Would purifying selection on existing genotypes give us pangenomes? So purifying selection would really discriminate between two different alleles. And when a new mutation came along and said, no, we've already got a really, really good allele here, so we're going to keep this one. That would mean that E. coli would end up with one perfect genome or close to perfect genome, and you only see a small amount of variation. So I don't think that purifying selection on really fit and really good genotypes, that's an explanation that's sufficient. What about drift? And so quite a lot of population biologists think that drift will actually be a useful and a good explanation for this. One of the things I didn't really say is that a lot of the accessory genes are private. They're unique to one genome, or they're maybe only found in 1 or 2 or 3 genomes. They're not really common. You're not finding 50% of the genomes have it and 50% do not. You're finding that a lot of the genes are quite rare. So there you've got a lot of core genes, you've got a lot of rare genes, and only a very small number of genes in the middle. So for population geneticists, this is really sort of saying to them, it's very, very likely that these are just genes that are transient. They're in the genome, sure, but maybe by next Tuesday, they would be gone from that genome, they'd be purged from that genome. And so they're really transient. And so perhaps random genetic drift could explain pangenomes. The next explanation is positive selection on new genotypes. So when a new allele comes along, so it's acquired by horizontal gene transfer, then natural selection says, yep, that's a really, really good idea, and we'll positively select that. So that obviously, again, would just give you a very reduced, sort of genetic variation, because it would say, yeah, perfect genomes and so on. But what about positive selection on new genotypes when the environment is constantly changing? That is to say that if you've got organisms like E. coli that can live in the gut, can live in soil, can live in air, can live in water, where there are lots and lots of microniches, perhaps natural selection could explain that. So new genes are quite often beneficial because it slightly shifts the ecosystem in which an organism can live and work. So I'm going to think about this. But there might also be an additional explanation, and that is to say that what goes on inside the genome could be having an effect, that genes having an effect on each other, and it may only be part of the story to think about genes fitting into their external environment. It might also be that they have to fit into their internal environment. Now, one of the things that makes us think that random genetic drift isn't really a very likely explanation for all of this variation. It is an explanation for a lot of variation in protein sequences, in vertebrates and so on, but perhaps not here. So these are two papers that I've been fortunate enough to publish. They're nearly 20 years apart. The top one is where we looked at codon usage in Borrelia burgdorferi, which is a bacterium that causes Lyme disease. And we see there really, really exquisite selection on particular codons and particular usages of bases in the genome. And now that's a very, very tiny, tiny fitness effect to say that you're going to swap out one codon for another. But it's sufficient that this organism has a preference for these. I'm going to take a look at this. Is again a bioinformatic analysis. It's a correspondence analysis, which is like a principal components analysis of codon usage in the bacterium E. coli. So you just take your bacterium, you take all of the protein coding genes, and you just split them into 59 numbers. Okay, a number for each codon, excluding the stop codons and methionine, which has no synonymous alternative. And so using these numbers, you make an array, and then you try to visualize that array, reduce the dimensions of the array using this correspondence analysis, this multidimensional reduction method. And essentially here, you've got the two axes on the X and the Y axis. The X one describes the greatest amount of variation in codon usage. And the genes that are in that oval, they're the highly expressed genes. And they use a particular subset of codons because natural selection has favored a kind of co-evolutionary process where in the cytoplasm of E. coli, you don't have all of the tRNAs. You've only got some. You've got fewer than 40, right? And they prefer to translate particular codons. It's a very, very slight difference because, you could have the same tRNA will decode UUU and UUC for phenylalanine, but it probably has a preference for UUC. Those tiny, tiny preferences in an organism like E. coli, which is a giant effective population size, that's enough. That's enough. Natural selection means that these proteins use the preferred codons. Even if the non-preferred codon still encodes the same amino acid, it uses the preferred one. So natural selection is really, really, really efficient in organisms like E. coli. So why does it have so much variation in its accessory genome? How can it tolerate all of that variation when in its highly expressed genes, it doesn't even really like to use a non-preferred codon in one codon of one protein of a 5,500 protein genome? The answer we have proposed is that this extraordinary amount of variation is selected because E. coli is very good at selection, at natural selection. It's very efficient. It's selected, and it's selected to allow different ecosystems to be colonized. And some genes will be really, really fit in some ecosystems, maybe not in others, but that's still sufficient for E. coli to maintain across its pangenome a very, very large collection of genes that on the whole, maybe not all of them, but for the most part, they're useful in one context or another. There's also the issue that within a given genome, perhaps these genes have preferences for each other. They want to live with some kinds of genes, and they really don't like to live with other kinds of genes. We developed some software to ask this question about genes and their relationships to each other. The program is called CoinFinder, and it's used to detect significant associations between genes and also what we call dissociations. So this is, if you like, war and peace within a genome. Okay, so we're really getting very far away from tree-like evolution here. We're getting to where we think that within a particular genome, there are conflicts, there are cooperations, there are compromises, there are all of these things happening. And what we're trying to do is to understand the mechanisms within a genome that allow this to happen, and more broadly in the pangenome, and then how all of those sit in the varying ecosystems in which they find themselves. So it's a bioinformatics crowd, so I'm going to go a little bit through the pipeline very, very quickly. We take whole genomes as an input, hopefully some hundreds of genomes, because this really works well when you've got lots and lots of genomes. Predict the open reading frames, cluster the orthologs, in other words, make gene families as best we can. We've got some filters going on. We filtered out the really low frequency genes because they can't really tell us an awful lot. And so we end up with a whole bunch of genes, and we ask, when we look at the pattern, their distribution across the phylogeny, across the organisms that we're looking at, are they showing a pattern of association? Are they showing a pattern of avoidance? What kind of patterns are they showing? And we've got a whole bunch of filters that they go through. And in the end, we make this kind of network, a different network to the ones I was showing you earlier, where the edges say, we really like to live together, or we really don't like to live together. So we've got an association and a dissociation network, and I'll take you through what this looks like. Okay, so this is pneumococcus streptococcus pneumoniae. We took a look at a bunch of genomes here. Here's the phylogeny here on this side. It might not be all that clear. And I've just focused in on a system of VATPases. They're a complex that are important for pumping things in and out of the streptococcus. Sadly, this has been colored in two different reds, but the reds over here essentially are the known VATPase genes, okay? And the standard software that we used to predict them didn't actually predict all of them. It predicted most of them, but it left out 3. But by looking at the presence and absence and the co-occurrence of these genes, we said, you know what? It's very, very likely that these other genes are important. So when we went to the literature, we found out that in fact, the original software hadn't picked these up as parts of the VATPase complex, but by looking at the distribution of presence and absence, we found that. And this makes sense. If they're found together, they work together and they produce a functioning VATPase complex. So that's fantastic, right? So this is just a bioinformatics program that doesn't know any biology. It's looking quite simply at which genes like to hang out together, which genes do not. It found this bunch here, and it had some known ones, and it predicted the others to be heavily involved. And when we dug in further, we found that in fact they were. But very, very interestingly, it's also predicted more than 50 other proteins that have a pattern of presence and absence that is far more similar to these VATPases than you would ever expect by chance. It's a smoking gun analysis. It's not direct evidence, but it is very, very suggestive that these genes here, they either like to live in this environment where like to live in a genome where these other red genes are present, or they're helping out with this whole VATPase activity, or VATPases make it really possible for them to live in the genome and do whatever it is that they want to do. Cystic fibrosis lungs are quite often colonized by Pseudomonas aeruginosa, which is a kind of opportunistic pathogen. We got over 200 Pseudomonas aeruginosa genomes from a particular study in Canada and CF patients in Canada. And each of these columns here are colored differently to represent sets of genes that show a pattern of association with each other that is stronger than we expect by random chance using all of the various statistics that we use. The network here is a network of these associations showing that. And so what we're seeing is again and again and again, communities of genes that if one is present in the genome, it strongly predicts that another one is present. And quite often, this can be extended across 4, 5, 6, 7 genes. And this is something that we see. We looked into the CF data and we looked into lots of other kinds of data sets that were there. And we said, well, are they expressed together? Are they more likely to be expressed? And we found that these co-occurring genes were far more likely to be co-expressed with each other. What about, sorry, I can't see here. Yeah. Do they have protein interactions? And it turns out that they're 7 times more likely to have a protein interaction than if we took 2 random genes that are in the same genome and said, do you have an interaction? So this pattern of trying to find out from presence and absence whether 2 genes like to be together or not only does it show they're more likely to be expressed when they are together, they're also more likely to have an interaction when they are together. And slightly surprisingly for us, they're more likely to have a Go annotation, which is a gene ontology. So broadly speaking, and I'm going to be slightly wrong here, broadly speaking, they do more or less the same thing, or they sit in the same kind of place in the cell and so on like this. So they're much more likely to be like that. And maybe it's because they're trying to do the same thing or operate the same function. This is starting to feel to us like we're getting to where we can predict genomes, which would be remarkable if we actually could in the future predict genomes. We certainly can for some small subset of genes have some sort of idea of what other gene might be present in a genome. And that's very, very interesting from all kinds of perspectives, like, understanding synthetic biology and antimicrobial resistance, defense mechanisms and so on. It led us to where we are today. And so this is the work that I'm actually doing today. Everything else up to now has probably been published, but this is where we are today. We're beginning to look at pangenomes in the same way that zoologists, for instance, might look at an ecosystem, a savanna or a swamp or something like this. And we're starting to think about the way in which genes have influences on each other. So earlier on, I was talking about sort of maybe, genes are beneficial in an ecosystem, but maybe genes are beneficial in a particular genetic background. And maybe there are places where they don't do so well. Think about it for a second. I mean, if you've got methanogens, okay, methanogens, they don't grow in organic compounds, they grow anaerobically and so on like this. If you put a sugar transporter into a methanogen, it doesn't use sugars, right? So that genetic background is just no good for that sugar transporter. But what about something much more subtle than that? So we're beginning to investigate pangenomes for things that ecologists look for, when they go doing field work and looking at macro ecological systems. We're looking for mutualism. What about if we find 2 genes and the presence of one strongly predicts the presence of the other, and the reciprocal? They each predict each other, and we're finding lots and lots of those. What about commensalism? Okay, so where the presence of one gene strongly predicts the presence of another gene, but the second one doesn't care. It's just not, it can be present in lots of other genomes and it doesn't care, but the first one really is only found when the second one is already there. So one facilitates the presence of the other. So, I mean, we all know about commensalism and, bacteria grow in your skin. So if there's no human, there's no bacterium, right? Competition. I'm going to give you a little example of competition in my next few slides. And the others are predation, parasitism, and asymmetry. And so we do know there's lots of parasitic transposons and other genetic elements that try to parasitize bacteria. So we're beginning to think about pangenomes, not just anymore of counting genes, but looking for a mechanism for why they really exist. This is just a little toy diagram. I'm going to go through it very quickly. Here you've got a group of organisms. Some of them live in the blue environment, Y, and some of them live in the green environment, Z, aerobic, anaerobic, you can think of it. There's a bunch of genes that are here simply because they've been inherited vertically down through the years, down through the generations. There's a bunch of genes here the same way. There's a bunch of genes that are present in the blue organisms because they would do well in the blue ecosystem. Bunch that are in the green organisms that do well in there. But then there's a bunch of genes, and these are the ones that we think that are very interesting, whose presence or absence is conditional on other genes in that genome. So the yellow to orange to red, or whatever colors these are, show a dependency. O is only ever found when M and N are also present. So those three examples there. The orange one is only ever present when the yellow one is already present. But the reciprocal is not the case. The yellow one is happily here and here and here and here and here, but the other one's not present. So there's an asymmetry in what we see there. And I'm trying to see the rest of my slides. Here you're seeing a kind of antipathy. So wherever you see the yellow one, you don't see the purple one, and vice versa. Okay, so we're beginning to think about this, but I guess your question is, well, do you see these kinds of things? So we published this a little while ago. It's with Nadine Ziemert in Tübingen in Germany. She's got a bunch of, these are halophiles here, and they produce siderophores, which are iron chelators, which help them to live in, the sort of halophilic, the salt-loving environments that they like to live in. But you look across these three species, okay, they're called Selenospora, and they've either got one or they've got the other of these gene clusters. The gene clusters here, the red one here and the gray one. It doesn't matter what their names are. The chemists are very involved, but for what I'm talking about today, it doesn't really matter. But there's three horizontal gene transfer events, an initial acquisition and two later horizontal gene transfers of this SLC, it's selenochelin, that does the same job as the ancestral type, the DES one. And so because they're the same, or they're producing the same kind of product, you either have one or the other. So this means that both of these are accessory genes and they're competing with one another. And so this is classic sort of ecological sort of thinking that might sort of make you think that, this might be common in pangenomes. Another one here is, this was done by Rebecca Hall in my lab, and looking at E. coli, and she's looking at the various systems where genes like to be present together at the same time. These pink ones are all DNA, RNA replication, regulation, and repair genes. And so this is a group of them, this is a group of them, this is a group of them, and down here, this is a group of them. The groups like to be together, but they're not with the other groups. Okay, they form their own community. They're a bunch of genes with different functions, but they're broadly speaking all DNA, RNA replication, and they're not really swapping out. These work well together. They form a kind of a coherent community within this E. coli pangenome ecosystem. What we're also finding out is that when you look at these pangenomes, and these are a bunch of phosphotransferases, sugar transporter complexes, and they're much more mix and match than people thought when we look at presence-absence data. A lot of these are known from experiment, where they're known that they work together. These three proteins work together, these three proteins work together, these four, etc. But these edges show connections where these are significantly co-occurring. So what we think up to now, that this might be, the phosphotransferase, this particular one here, it turns out that, yeah, okay, maybe these are to be found together, but several of these proteins, these two here, can also be found associating with these other ones. So these are mixing and matching and sort of, maybe moonlighting in a different way. So the last thing I want to talk about is how we're really thinking about this ecosystem model, if you like, in a more sort of complete way. And we're using a random forest approach. And so random forests are a sort of part of, I'm just looking at my watch, I've got 5 more minutes, and that's just about right. I hope I'm not talking too quickly. I hope some of this is making sense. But so random forests, right? So I tell everybody random forests, they're kind of machine learning approach, and I make sure that people know it's machine learning, because right now that means that my science is great, because machine learning is the best right now. But random forest is very simple. It's a really, really simple idea, but it's really beautiful as well, I think, you know, in its elegance and simplicity, and how it helps us then as well. We're asking the question, up to now, we're looking at pairs of genes and whether they have a pattern of presence and absence that sort of like, you know, means that we think that they hang out together a bit more than you expect by chance, and therefore they might be functionally related. And when we look at the functions, we find that's true for those things that we have functional information for. So the random forest approach is trying to do something more subtle, but also more complex. Okay, the first method just did pairs of genes. This one does the following thing. So we pick gene A. Our objective is down here. Does some combination of gene presence and absence predict the presence or absence of gene X in my genome? So we start off and we randomly sample gene A. And we say, okay, partition the genomes. These ones have gene A, these ones do not. And then we move down along and we look at gene B. So these ones do not have gene A and they do not have gene B. And oh, look, they've got gene X. Down this side, these have gene A, they have gene B, they have gene D, and look, they've got gene X. And so this is a toy example. We make our decision tree. This is a decision tree. We make it much longer. It's got a depth of about 8. This is a depth of 4. So depth of about 8. And we ask, for every gene in every genome, is there some combination of presence absence that predicts the presence of that gene in the genome? And it turns out that for a lot of genes, yes, their presence or their absence is predictable. We eventually make a network like this because quite often we're finding, again, a community of genes that kind of predict each other, the mutualisms that I was talking about, but there's also the other sort of commensalisms and antagonisms and so on, things that predict each other and do not predict each other. This is a decision tree. It doesn't matter. I'm not going to get into it. You're a bioinformatics community. Go take a look at this. I'm sure you're more than familiar with this. A decision forest or a random forest is where we do lots and lots of these decision trees for every gene. We do about 1,000, and we ask whether a gene is predictable and what other genes are capable of predicting it. And this is where we're moving today. This is the E. coli pangenome, and we've only included the genes in here where they have at least something that either predicts their presence or predicts their absence. So if there's a strong prediction of presence or absence, I've included it here. It's not every gene, because even with just 500 genes, E. coli genes, that finally becomes an awful lot of genes, and it's very difficult to see it, but we've got some here. And I'm just highlighting a little group here. And this group here, they really don't play well together. They don't like each other very, very much, okay? I don't have functions in here, and it's not the point that I'm trying to make about functions, but just that they exist, Group 36017, it's a gene family. If it's present in your genome, we can pretty much predict that this gene family will not be present. This one will not be present. This one will not be present. This one will not be present. And here's a reciprocal avoidance of each other, okay? So they really, really don't like each other here. They're not commensal organisms. And down here, we've got positive interactions. So family group 3562 predicts the presence of this one. Doesn't predict the presence of that one, but if this one is present, then maybe some of the others are present as well. But the arrows point to from the gene that predicts the other one, the predictor to the predicted. And as we build up larger and larger data sets, this is where we're going with this. We're borrowing quite a lot of ideas from, from language models and, that sort of machine learning. If I ask this audience here, if I said the word finish, can you tell me, or predict for me, what the next word in the sentence is going to be. If I said finish, how many of you would say finish up, finish down, finish first, finish last? And I've done this in classes, and people put up their hands. But what if I said to you, nice guys finish? Then suddenly almost everybody in the class says, nice guys finish last. It's a strong predictor of what the next word would be. So that's what we're trying to do with this sort of machine learning approach, to say that the presence of a gene, the absence of presence of gene A, absence of B, presence of C, presence of D predicts X. And those are the kinds of patterns. And it turns out that, and I haven't got the data here because it's only last week that we've got it, so I don't have good slides, and I don't really want to, I'll talk about it, but I don't want to put up a slide in case it turns out next week that it's wrong. But we feel that about 1/3 of the genes in large pangenomes like E. coli and the data sets that we have right now, about 1/3 of the accessory genes are predictable. Their presence are predictable. So for me, when I talked in the beginning about whether pangenomes were neutral or positively selected and so on like this, I'm afraid I'm very, very strongly coming down on the side of they're positively selected, or at least large parts of these genomes. The genes that are in there are there for a reason. And if we can build large enough pangenome models, like we have good, language models. I mean, if this is being recorded, the transcript from this will almost be perfect. Even with my strange accent, it will be perfect, right? That wasn't the case 2 years ago, but the language models have improved so much and have gotten so big that right now transcripts just are almost perfect almost every time that we look at it. These are a bunch of people that over the years have been very, very influential in both how I think about things and how we've done things. Bill Martin was very important for the eukaryote work. Mary O'Connell has worked with me in Pangenomes for a long time. Eric Baptiste on networks and thinking about things in a network way. These are additional people in my lab, but most of the work in this were the people at the beginning that I showed you there. That's the end of my slide deck. Thank you so much for inviting me here. I'm more than happy to take questions if you've got any. I'll stop sharing my screen right now and hopefully come back to you. Has that worked? Am I back? Oh.
Thank you, Dr. McInerney. It's very awesome. And I see in the chat, Katie Dylan had questions. Please, everyone, if you leave anything in chat or if you have questions, just speak out loud. Maybe we can start from Katie.
Yes, so shall I just read Katie's question? So, which is,
Yes,
you speak
please.
about variation like it is solely a negative thing. Am I naive in thinking that variation could be positive, negative, or neither? No, you're not at all, Katie. No, absolutely not. It can be both, and maybe I was maybe too negative about variation. So variation is of two kinds. We talk about pangenomes: gene acquisition and gene loss. And for the most part, I think change is probably a bad thing. But the changes we observe after a few generations, we can think about them. Positive variation, we've always thought was a little bit difficult. A little bit difficult to sort of get new genes that are positively selected. Now, but the vast majority of evolutionary theory has come from studying animals, okay? And so it's very, very difficult if you look at sort of some mammal or whatever, and it lives, it grazes grass or something like this, what enzymes would improve the likelihood of that animal passing its genes to the next generation? You're going to really search through the whole genome to find a new mutation that would really improve that. You might or you might not. So we've kind of had thought about new variations as being usually lost, unless they're neutral, in which case they come through. But positive selection on new variants is possibly the more rare thing. Now, that might not always be the case, but the one that I think that is possibly in organisms like E. coli, the most rare, is neutral variation, where you substitute some genetic element for another, one base for another, one gene for another, etc. Because, and it gets a little bit into the mathematics, the likelihood that you would be neutral is that if your fitness effect on the genome is less than 1 over the long-term, the NE, the effective population size. So E. coli's effective population size, 25 million. So the difference between the wild type and the new mutant has to be a fitness effect of less than 1 in 25 million for that to be truly neutral. Very difficult to substitute anything for anything else and have that tiny fitness effect. The vast majority of fitness effects will be greater in magnitude than that. So, and not just that, but it's a long story, but it has to be less than 1 over NE for NE generations. It has to be constantly like that for 25 million generations. It's very, very difficult. I'm not saying it can't happen, but I think that you can have transients, you can have genes that are in a genome for a short amount of time before they're kicked out, and you've just happened to sequence them, sure, but in the long term, I think it's very difficult for a gene to be neutral. I'm a big fan of genes being positive. I mean, I think a lot of this new variation is positive selection on sort of new ways of doing things, new ways of putting combinations of genes together. And that means that you can slightly live in a slightly different ecosystem or change in some other way like this. And bacteria in particular, they live in apart from things like Mycoplasma genitalium that lives just in humans, in sort of moist areas of humans. But most of them live, bacterium, E. coli, it's going to be in water today, it's going to be in soil tomorrow, it's going to be in a micro niche somewhere in the gut the day after. So there's a lot of variation that goes on there. So it's a lot of possibility for change to happen. So sorry if I said that variation, or if I gave the impression that it was negative. I just think that positive selection happens quite a bit. Negative selection. Yeah, if you try to lose a ribosomal protein, that's going to be selected against. I'm pretty sure. But in the accessory genome, I think positive selection on new variants is probably common enough, and neutral, I think, is probably rare. I don't know if that answered your question, Katie. Shall I go on to the next question that's here? Is this variation in pangenomes not a necessity in a rapidly dividing organism? I think so, yes. In the absence of some mechanism to maintain variation, one would end up with an organism that is extremely well adapted to whatever niche it happens to currently occupy. And when the selective pressure changes, the population can no longer have the capacity. And you see the amount of variation in pangenomes correlates with generation times. It correlates not so much with generation time, but how cosmopolitan the distribution is for the species, if you like. So E. coli, you find it in so many places, and there's so many micro niches that it could live on. Salmonella the same way, and so on, all the way down to intracellular parasites like Mycoplasma genitalium, which I've just mentioned, which just lives, it seems, we only detected in one or two places, and it's got no pangenome really. It's got almost no accessory gene variation. So the real axis, the biggest axis from big pangenome to little pangenome is how many niches and how many different places you're likely to find that species. Does it correlate with generation time? Probably does. There's probably a whole bunch of things that have confounding effects on one another, but for the most part, large effective population size occupies a bunch of niches, big pangenome. Is there a replication bias in the presence absence, plasmid or chromosome? So we're finding everything. We're finding that presence absence due to plasmid presence and plasmid absence is part of the story, but on the host chromosome, you're finding it the same as well. And in fact, the variation is, I think, nearly the same between the two. I think we haven't really enumerated or studied the numbers much, but it's everywhere. It's actually in plasmids, it's on chromosomes. You've got transposons, you've got all of those things contributing to it. Lots of the presence absence pattern variation is because things are in a cluster or they're in an operon, but just as many things are not. And in fact, there's some sort of very strange thing that genes that are very far apart are more likely to be found together in a genome. So they're far apart, but likely to be found together. So, sorry, I can't. You might be
before the next question.
Far apart in what sense?
Far apart is in when you measure the distance on the chromosome as you go around.
Really?
And they're
Okay.
quite a bit apart on the chromosomes where we find them, but they're found on the same chromosomes. So what that's about, your guess is as good as mine. But yeah, we find that too.
Yes, thank you.
Katie, Nathan, have you applied a natural language model to your data? No, Nathan, but be particularly excited to try doing that. Yeah, I mean, a lot of what I've just talked about at the end, it's not peer reviewed, so it's a way to see how much people like it. but I think the data is quite strong. And so we're moving into the space where we're trying to, steal ideas. All of the stuff that I presented here are ideas stolen from engineers, really, all the multivariate analysis and all of that. So natural language processing, it's all there. It's all in the TensorFlow library or whatever the Google support. You can use all that, and we're doing that, but I don't have results and data for that. But yes, it's so like a language, so like an ecosystem, whichever way you want to think about it. But it's not just tree-like evolution that we're looking at anymore. It's possible to have a genome stripped of all the accessory genome and yet viable. No, it turns out that people have tried that. And maybe 10 years ago, there was what they were calling Life version 2.0, and I can't remember, Craig Venter was at it. I'd have to go take a look. And so they tried deleting all of the genes that had no functions, and they were individually, you could delete them. But try deleting them all, and it wasn't viable. And so they finally have ended up with a stripped down genome with like 200 genes in there, or 150 genes. They didn't know what they did. They didn't seem to be involved in central anything, but you tried to delete them and they couldn't get the organism to live. So far the answer is no. But wouldn't it be really interesting to try and design a new genome? Talking about design and evolution is never a great thing, but anyway, but this is where we're going. This is, if genes are predictable, their presence or their absence is predictable in a genome by looking at the genome context, all the other genes, then yeah, we should be starting to be able to really think about context and subtle and complex patterns in these things. Okay, sorry. So there are hands up. Is that it? Benjamin, do you want to? Yeah.
Zari Hassan.
Yeah, thank you. That was a nice talk. So I was wondering, when you were talking about metabolic genes and tree-like history, and I think that you mentioned that the evolution of metabolic genes shouldn't be presented through a tree-like fashion. Could you care to explain why?
So I think my feeling about all of this is, don't start with a tree. Don't start with the assumption that everything has sort of like that you can make a phylogeny and every gene can be mapped onto that perfectly, because we know it can't. Many of them are compatible, that their evolutionary history for whatever group you're looking at, whether you're looking at alpha proteobacteria, all bacteria, whatever, they might have a very, very compatible evolutionary history, but others might not. So I think that you can come back to sort of using the tree diagram when you've got a big cohort of genes that have a similar or the same evolutionary history. But I think it's not good to start from that perspective, assuming tree likeness, when in fact all of these other sort of network-like sort of processes can happen. So I didn't say you could never do it, but I think it's probably more sensible to start by thinking or assuming that perhaps in your data set you might have extensive horizontal gene transfer, and if you don't, then a tree might be a great way of representing the data. But if you have lots of horizontal gene transfer, then the tree, I don't think, really works.
Just a small follow-up question. So, I mean, when you are presenting this in a network model, I mean, I found it pretty fascinating. At the same time, I mean, as I'm not used to those kind of models, it wasn't visually that pleasant to me. I mean, it was great. I mean, networks in general, I mean, I think that there are a lot of things going on. I mean, I'm more used to the phylogenetic approach and stuff.
Mm-hmm.
So the first thing is, are you trying to come up with new models of presenting evolutionary history other than network models? The first question. And do you get that a lot, that the network models, I mean, I'm thinking that the network models are not that visually pleasant to me in general, but do get that a lot from people who are first to see that, I mean, this kind of model can also represent evolution and stuff?
Yeah, so I think it's a mixed bag in terms of some people are really unhappy with leaving phylogenetic trees alone. But my problem with using phylogenetic trees only is that I think it masks process. So if we're all interested in the evolutionary history and the process that led to the patterns that we see today, the organisms that we see, and typically that's what people are interested in. How do we get to the data we see today, and how do we make sense of that? So if you're not representing it properly, then you're going to make an incorrect statement of the process. I'll give you an example. So all of us here, we've got two parents, right? If we said we're going to have to use a phylogenetic tree, so let's use some outgroup, another primate as an outgroup, and there's me, my mom, my dad, right? And I have to make a phylogeny, right? Then there's only three possible topologies. It is, assuming the outgroup, that I'm the most closely related to my mother with my dad outside. My dad is more closely related to my mother than either is to me, and that's the offspring early hypothesis. Or the other one, I'm more closely related to my dad. It doesn't work. What works is a pedigree. It's a different figure. It's a different structure, right? A phylogenetic tree would not work. We could make it. We could go and get, genomes. We could download them from NCBI and make a phylogeny, force the phylogeny. And so what I'm saying is that phylogenetic tree making software will do what it says. It will make a tree. My question is, should you? Just because you can, maybe you shouldn't. You shouldn't. And I agree with you about the network. So I put them up, right? And they're big and they're just a big fur ball and there's nothing there. What we really need to do is to go into those networks. And I kind of maybe showed in one of the figures where we're looking for motifs, things in the network, parts of the network, because the whole network with 5 million sequences, I think one of them might have had. It's too much. It's not going to educate you. So what we were really trying to do is, for instance, going into the VATPAs, as I pull it out, and my figure had a phylogenetic tree and a network. Phylogenetic tree is a kind of backbone phylogeny, but you can see that the distribution of the presence absence of the VATPAs is that wasn't. It wasn't in one clade. It was distributed across with lots of acquisitions and losses in there. So I think in the end, if you want visuals, you can use a mixture of two. You're correct. The big network is just difficult to see, and you don't feel you've learned something except that there's a lot of stuff happening. But you can dig into it. You can use things like Network X, which is a software library. You can pull out motifs off that and sort of say, you know what, for these 7 genes or these 200, there's something interesting here that I want to explore a bit further. So I can't say I disagree with you on all of these, but I'm still going to say that the ways of thinking about and ways of depicting and ways of analyzing. So we want to do two things: visualize and analyze these horizontal gene transfer events and understand what they mean. I think those are in their infancy right now, and maybe in 10 years' time, we'll have software and ways of doing things that are just as good as the phylogenetic software that we have today.
Thank you.
Olivia.
Okay. Hi.
Hi.
Thanks so much for your talk. I really liked it. It kind of reminded me how, this is a comment before I do the question, but how a lot of ways that we think about biology is really metaphors. And if you kind of deconstruct what the metaphors are that we're using, then it can kind of remind us that, a phylogenetic tree is a metaphor. It's not necessarily real. So we can think about things in different ways. So I like thinking about stuff like that. So it's really fun to hear your talk. But I do kind of have a question about sort of about like definitions of what would constitute a pangenome and how that would relate for poorly defined species. So like E. coli, even if it's very diverse, we have a very distinct idea what it is, and we have a lot of information on it. But how would this approach work for species that are poorly defined and have little data? Like how much data and information about something do you need for this approach to be useful?
Okay, ever so slightly controversially or whatever, I actually think that the pangenome is really just a sample. It's so when you sequence the first E. coli genome, every gene is core. When you've got 2, probably a giant core and only a very small accessory genome. And all that happens every time you sample a new genome is that the core tends to get smaller and the accessory tends to get bigger. Where you stop, nobody knows. We're actually doing a pangenome analysis of all the halophiles, so several genera and so on. So for me, they're just samples. I think there's a little bit of a difference in the sense that the core genome and so on can undergo recombination more easily, you know, using the REC-A and all of these methods for holiday junctions and so on. And when they're a bit more diverse, you're dependent on some kind of mobile genetic element to bring things in, and you've got illegitimate recombination. Nonetheless, I think all of these conflicts and compromises and so on happen across all scales. So for me, I'm probably less worried about this. E. coli is, of course, a terrible species because, I mean, E. coli and Shigella are, you know, Shigella is, what's the phrase? It's E. coli with a little coat of toxins. I'm going to get that wrong ever so slightly. But, you know, Shigella and E. coli are not different species. They're, you know, Shigellas arise at least 3 times from within the E. coli, you know, diversity. So I'm probably less worried, but maybe I should think about it more deeply. Has that helped to answer the question, or just confuse things more?
Dr. Kenneth, do you still have questions?
Yes. Hi, James. This was interesting talk.
That's okay. He answered my question. Go ahead.
Sorry. Okay. This was interesting talk. I was wondering if you have, if you can elaborate about. So when you talk about horizontal gene transfer, obviously the 2 bugs were closely next to each other and gene was transferred. If you are taking population today, let's say the tooth biofilm, and there are maybe 7,000 bacteria, how many of them are shared same genomes based on the horizontal gene transfer, or you cannot find them anymore and they are dispersed?
Okay. Yeah. So I think this is an interesting thing. And Jesse Shapiro in Canada is working on microbiome stuff to try to find. You're talking about within microbiome horizontal transfer and how often it happens and that sort of thing.
What I'm saying is, once you're done, are you still in the same environment, or and this is a way to explain it, or you are disappear from the environment going elsewhere?
I actually don't know the answer to that question. To be, if I'm to be absolutely honest, I don't know. You know, there's an awful lot of naked DNA that sort of persists for a while, and you know, so I don't know the relative influences of active transfer. I mean, you've got DNA uptake by some organisms. Are you gone when you die? I don't know.
I have just another question. So you mentioned that there is a core and accessory genome. Do you think that the rate of exchange of nucleotides is similar between the, if everything is random, like you are proposing, is the rate of randomness between core and accessory genome is the same?
Okay. First of all, the rate of random acquisition and loss, I think, is the same, but retention, I'm saying that natural selection is retaining genes that work well together and is preventing the retention of genes that work badly together. So it's not that I'm saying it's random. You know, I think, you know, genes can come and go at random, but natural selection is sort of saying these work well together and they're not, and that's what structures pangenomes. Between the core and the accessory, in terms of the per nucleotide exchange, my feeling is that the accessory genome in many so first of all, actually, in answer to another question, I think there's a whole gradient from organisms like E. coli. Neisseria has a huge amount of recombination within the species, all the way down to mycoplasma, which is very, very little. Okay, so I think that the answer will be different in different organisms, but overall, I think that the accessory genes are far more likely to see recombination and transfer than the core. And there's some work done by Tal Popko from some years ago where he talks about genes that encode proteins that have lots of interactors. They are horizontally transferred far less often than those that have very few interactions. So he felt that was the major discriminant between them. So in that case, then of course, things like ribosomal proteins are rarely horizontally transferred at all. Whereas something like a protein that sits on a membrane and just does one activity on its own, that can probably be much more easily transferred. So my feeling is that the accessory genome undergoes more horizontal transfer, but it would be really interesting if people updated the numbers and had a good look at that. Yeah.
Thank you. Thank you.
Dr. Ann.
Yeah, thanks. I'm going to ask a question that is way outside my wheelhouse, and that is, once upon a time, there was the Cambrian, and it exploded genetically and in lots of other obvious ways. And I don't think we have any way of looking to see whether or not tempo was an issue here with respect to these kinds of changes. Was there an explosion in from a core genome to a larger genome? And we can't ask that, but I saw a paper recently, and I can't remember exactly where it was from, but it had to do with noting variation sort of increasing in a certain plant species as they move to safer territory with respect to their temperature tolerance. And if we're going to get through this any faster than the Cambrian, we're probably going to need something like horizontal gene transfer. And is it possible to look for that in deliberate ways, or think about it in the kinds of ways that you are talking about bacteria now?
The answer is yes. And so I had one slide on halophiles and their acquisition of genes from bacteria. And we think that the majority of those genes all came in at the bottom of the halophiles, and whoosh, you had a methanogen rapidly transformed into an anaerobic chemolithotrophic methane producer into an aerobic heterotrophic thing that likes to do photosynthesis and so on, and that probably happened very, very quickly. And we're still looking.
That wouldn't be coeval with the Cambrian, right? This is much earlier.
No. I think that's got to be 1 billion years ago, maybe a lot longer, and the Cambrian is much more recent. Yeah. But I was very fortunate to meet Dick Lewontin once, and we had a chat about punctuated equilibrium and all of that, and he said that he was very cross with Stephen Jay Gould, who always presented it as like steps, straight up, lots happened and then nothing, and then lots and then nothing. And he said it was just like rate variation, as it went along. That was really what they thought that they saw in the fossil record for that. But I think that there's a lot of what also happens in bacteria and archaea, and I think we possibly haven't looked enough at it. And I think, so one of the things that I didn't actually talk about, I was hoping to, or it's in our thinking, is, keystone species, right? So classic ones are, when beavers set up dams and so on like this, and then all of these other animals can come, and plants can live in the lake that's formed from this dam. And so the beaver is a keystone species there, and wolves are quite often keystone species. But we think they're keystone genes that when they come in, they facilitate a whole bunch of other genes that can now live in this genome that previously might have been hostile to it. Or if a gene is deleted, that it makes way for these other ones. So these turnovers that you talk about, we actually think that we see them. Now, I didn't present that, but we talk about it in the lab, because we think that we see this in the data as well. These keystone genes have a massive influence in lots of other genes, and this gene, might be sort of, if it's present, then 30 other genes can be present in that genome. If it's absent, they can't, and that's that sort of thing. So what you're talking about, give me a couple of years. I think it's in prokaryotic pangenomes as well.
Oh, thank you very much.
Can I ask a question? So I don't know how to raise my hand. So we actually work on the we don't work on the microbial genome, but we work on the canine genome. I was thinking using the pan genome, because the dog really has I'm working on the canine genome. There's like 300 to 400 breeds. So can we apply the same concept, try to understand it? For example, using the wolf as the core genome, and then try to understand how each breed was evolved.
I think so. I think so. And so I don't do so much in sort of big eukaryotes and so on, but there's a lot of literature on intragenomic conflict and then that sort of thing. So I'd wonder if dogs are, of course, a wonderful, model organism to work on because of all of that variation. But I wonder as well if there are SNPs, maybe not so much gene presence absence, but SNPs that are just facilitating so many other things. And if you've got this particular SNP, you've got one set of other things that are possible, and so on. So I think I feel that this kind of way of thinking can sort of be used elsewhere as well. I mean, again, we've just stolen it from the ecologists and thought, well, could genes be analogous to species on a savanna, or in the ocean or something like this? And again, all the way down to SNPs. And I know the population geneticists feel that there's a lot of intraspecific variation where a mutation really has a lot of influence on other mutations that can or cannot happen. And you've got epistasis and all of those interactions as well. So the answer to your question is, yeah, I think that thinking about in terms of mutualisms and antagonisms and so on. I think that could be a very profitable way of doing that.
Yeah, that's another thing is the cancer genome. I mean, the cancer genome people, it's another. I think this is a really, really, really powerful approach. So I really enjoy your talk. Your talk is just fantastic.
Thank you. Thank you.
Thank you.
Great. Any other questions? If not, then I think that's today's seminar. Thank you so much, Dr. McInerney.
Thank you so much for inviting me. It's been a real pleasure. Thank you.
Awesome talk. Thank you.
Yeah. Thank you. Thank you for sticking around and answering all those questions.
That's all right. That's fine. It's perfectly fine.
All right. Have a great weekend.
Yeah, you too, guys. Have a great weekend. Okay. Take care. Bye.
Bye.
Bye.